Hello and welcome to our last video in our Chapter 38 video lecture series, where we are going to talk about muscle contraction and locomotion. In this section, we want to focus on learning objectives here to classify the different types of muscle tissue and explain the role of muscles in locomotion. So first, we need to discuss our muscular system. It's going to function in the movement of an entire organism and movement of materials within the organism, whether it's going to be blood or food. So the three types of muscle tissue that we have, and you may remember this from our tissues lecture, is going to be smooth muscle, cardiac muscle, and skeletal muscle. And the cells within these muscle tissues are going to be called muscle fibers. So we can use this term muscle fiber interchangeably with muscle cells. First, let's talk about skeletal muscle. We'll see that the cells here are going to have a tubular, multinucleated, and striated appearance due to the myofilaments that we have within here called actin and myosin. Actin is going to be more of a globular contractile protein that will interact with our other myofilament called myosin to allow for muscle contraction to take place. We're going to talk extensively about these two proteins in the coming slides. And then I want you to look at this illustration here. Notice that these cells have many nuclei within them. That's because these muscles are going to be very long and attach from one part of the skeleton to the other. And so because this muscle cell can be so large, we need to have multiple nuclei to kind of be the control center and also allow for protein synthesis within the muscle to take place in multiple locations. Now, skeletal muscle is under voluntary control, meaning that with conscious thought, we're able to contract these muscles and cause movement to take place. Whereas with smooth muscle and cardiac muscle, it is involuntary. So let's take a look at smooth muscle next. We're going to see that the muscle cell or muscle fibers are going to be shaped like cylinders with pointed ends. Sometimes they're referred to as a spindle shape, where they're going to be a little bit thicker in the center and then tapered out at the ends. These cells are nucleated, meaning that we only have one nucleus per cell, and we see them in parallel lines that form sheets. We don't see striations here, and that is because we are not going to have the same exact arrangement of proteins here as we did with our skeletal muscle. The areas that we're going to find smooth muscle in is located within our hollow organs like the stomach, the intestines, and also our blood vessels, so that we can have these walls contract. But again, this is involuntary. It's with our unconscious thought that this takes place. Another thing to note is that our smooth muscle is slower to contract than our skeletal muscle. So it can sustain prolonged contractions and doesn't fatigue easily. All right, for our cardiac muscle, we're only going to find this in the heart wall. Our fibers here are uninucleated, so we only have one nucleus per cell. And here it's going to be striated just like our skeletal muscle and has a tubular shape to it. We'll also see that it is branched. So you can kind of see this over here where we have some branching taking place of the cell, and they interlock with other cardiac cells at these areas called intercalated discs. And in these discs, we're going to have special gap junctions, so almost like pores that are going to allow contractions within one cardiac muscle cell be transferred into another one, so we can have parts of the heart contracting at the same time. For instance, the atria contracting at the same time, and then our ventricles contracting at the same time. We will also have these cells relax completely between contractions, which helps these cardiac muscle cells prevent fatigue. When we contract, it's rhythmic and it's going to occur without nervous stimulation, because we will see later on in MP, you're going to learn that we have these non-contractile cardiac muscle cells that sit in an area known as the SA node that is going to set the nerve contractions or impulses moving through the heart to cause it to contract. So this is involuntary. So we're going to spend some extra time talking about a skeletal muscle fiber. First, we need to understand the structure of the cell. So we don't think of the muscle cell as being a round, typical cell. Instead, we know that it's cylindrical. So its sarcolemma, which is its plasma membrane or cell membrane, is going to be along this area here. Notice how it kind of encircles these fibers on the inside. The sarcoplasm is the special name for the cytoplasm within the cell, so we would find that right along in here. And then our sarcoplasmic reticulum should sound familiar because that is going to be the endoplasmic reticulum of our muscle cell, and it's a special site where we are going to store calcium. We can actually see a better view of what this looks like on this slide over here. And it is going to be this area that we see in the purple, kind of branching out along in here. So notice how these first three terms all start with the prefix of sarco. Sarco means muscle, and so we know that these structures belong within our muscle cell. Another unique feature is our T, which stands for transverse tubules, and these penetrate the muscle cells and come close to the portions of the sarcoplasmic reticulum. Again, we see that a little bit better in this illustration here. The purple was the sarcoplasmic reticulum, and these T tubules kind of extend between them here. And so the calcium is found within the sarcoplasmic reticulum. And so it's when we get a nerve stimulus to the muscle cell that will move down the sarcolemma and eventually enter the T tubule that will then signal to the sarcoplasmic reticulum to release its calcium into the myofibril area. Now within the sarcolemma, we are going to have a lot of myofibrils, and these are going to be the contractile parts of our muscle fibers. So our sarcoplasm in the center here will contain glycogen, which if you remember, glycogen is a larger molecule of a bunch of repeated glucose molecules. So we can always break glycogen apart and use that glucose to provide energy for muscle contraction. The sarcoplasm will also include a red pigment protein known as myoglobin, which is going to bind oxygen, and we can utilize that to create more ATP. So let's go back to our myofibrils here within our muscle cell. Like we said, we are going to have many myofilaments in here that are essentially our contractile proteins, and they are going to be assembled into larger cylinders so that we can organize the proteins here for muscle contraction. And that's what essentially creates these striations based on the placement of these myofilaments. So we have those two different types. We have our thick filaments, which are known as myosin. And in this image over here, you could see that they are the structure in red here, whereas the actin filaments are thinner, and you could see them in this yellowish orange color here, attaching to what's called the Z-band, and they are going to overlap these myosin filaments just a little bit, and as we contract the muscle, we will have even more overlap. So the actin will move closer to this area that is in the center of the H zone, and we call this line here the M line. So notice how if we go back to this Z-band over here, we see we have another Z-band on this side. This space in between consists of what we call a sarcomere, and this is going to be our functional and structural unit of our muscle cell. And if you look at this myofibril or muscle cell here, we see that we have repeated sarcomeres throughout the myofibril. Now let's take a closer look at that sarcomere. So as I said, a sarcomere extends from one Z line to the other here. And this is going to create a dark band on either side. When we look at a lighter colored band, so let's go back over here, where we have this I band, notice how the Z line is kind of in the middle. And the reason that we call this I band the light band is because we are only going to have our thin myofilaments here, the actin filaments. Whereas the A band, which we see pictured over here, is going to be called the dark band. And this is called dark band because of the overlapping of our actin myofilament over the myosin myofilament, and therefore it appears darker. Now, in the center within our A band, we are going to have a vertical H band, and it's not pictured as well over here, so I'm going to flip back to this one, but that's going to be where we have well, I can't say no myofilaments, but we're going to have only the base or the center of the thick filaments. So it is going to appear just a little bit lighter in there. And in the center of that, we are going to have that M line. Let's take a look at the structure of that thick myosin filament. So when we look at it, the molecule is shaped like a golf club, and the rest of it is going to be straight. And what we will have happen is that these myosin heads will attach to actin and eventually shift the actin molecule toward the M line. So these blue actin molecules would move in toward that M line, which would cause the sarcomere to shorten. Now, and let me also just mention that when this globular head of the myosin protein attaches to the actin molecule, this is called a cross bridge. Makes sense, right? Because we're creating a bridge here across the two proteins. So when we look at our actin filament or our thin filament, you're going to look at these round yellow globular structures. That's our actin here. And it's going to be intertwined with other proteins. This spaghetti noodle looking like protein here is called tropomyosin. And then the green globular structure here is called troponin. Now these are very important molecules, or proteins I should say instead. So what happens is when calcium is released, and I'm going to go back here. Remember, calcium is stored within our sarcoplasmic reticulum here. When we get the proper signaling, we are going to release this calcium from the sarcoplasmic reticulum and will reach the region of the actin myofilament. And on the actin myofilament, we had troponin. Now, troponin binds to three different things. One, it's going to bind to our spaghetti, our tropomyosin. Two, it's going to bind to the actin molecule. And three, it's going to have a spot for calcium to bind to. So in this video here, we could see calcium is moving down this pink dot and attaching to troponin. When calcium attaches to troponin, it causes this tropomyosin rope-like structure to shift. Now we want that shifting to take place because when our muscle is at rest, we have these active sites that are covered up so that the myosin head cannot bind to actin and create a cross bridge. But when we want contraction to take place, the calcium binds to troponin, which causes a shifting of tropomyosin, revealing the active sites, and the myosin head can bind to actin and cause a contraction or a shortening of our sarcomere. So this is what that looks like. The myosin heads are attaching to the active site and moving the actin filament toward that M line. And it's going to do this over and over and over again in order to create force and tension within the muscle. And this is a picture of what is happening in the sarcomere. So this is our sarcomere from Z line to Z line at rest. When we create those cross bridges and we're bringing the actin filament in toward the M line on both sides, we are going to see a contracted sarcomere. So here we have complete overlap of the actin myofilaments with our myosin filaments. And we could see we have smaller I bands as a result, whereas our A band is going to stay consistently the same. We also have a disappearing of that H zone. Remember that H zone is where we only see the myosin myofilaments, and here we don't have that anymore because actin came in toward the center. And so how are we going to have myosin attached to actin? We need to power it and utilize ATP. So let's go through the cycle of how that takes place. So first, we are going to have ATP split when the myosin head is unattached. So it's moving back down, and it splits into ADP and inorganic phosphate. We see that ADP and inorganic phosphate are bound to the myosin as the myosin head attaches to actin when those active sites are revealed. And when we release our inorganic phosphate, the myosin head is going to create a power stroke. It's going to pull that actin toward the M line. And then we will once again bind ATP, and that allows the myosin head to move back down into the resting position. If we are going to have it create a cross bridge again, we will split ATP into ADP and inorganic phosphate again, and the cycle can rotate through one more time. All right, so like I said, we'll have this process take place over and over again. Now, when a person dies, we are going to have their body in rigor mortis, where we no longer have ATP available, and we know that we need it in order to get the muscles to relax. So the body becomes very stiff, and if the body is there long enough, we won't have ATP present to get the myosin head to detach from the actin, but instead what will start to happen is protein breakdown. So eventually the body will become relaxed, but it's not because we have ATP present. All right, next let's talk about excitation contraction coupling. So we need to describe a structure called a motor unit. And this is when we have the motor nerve fibers coming from our spinal cord and supplying muscle fibers that it innervates. So when we have a muscle, it's not just one axon that moves down and supplies all of this. Instead, we have these separate different motor units, and this is going to be really useful for when we want to use our hand to pick up a pencil versus when we are using our hand to pick up a 50-pound dumbbell, right? So that way, when we're picking up a pencil, we can only utilize a few of these motor units so we don't create too much force. Whereas when we're picking up something heavier, we are going to utilize more and more motor units to be able to do that. And we'll recruit more muscle fibers so that we can create more strength to pick that heavy object up. Now, next we're going to talk about the very end of this neuron, that axon terminal. So when we look at this image here, there's the axon terminal, and it is meeting the sarcolemma here. If we take a closer look at this neuromuscular junction, we're going to see that the nerve is going to sit closely to this folded up area of the sarcolemma, and this is called our motor end plate. When we get a signal from this nerve to make the muscle contract, we have those neurotransmitters. You can see a close-up over here. The neurotransmitters within the synaptic vesicle released within this space called the synaptic cleft, and those neurotransmitters are going to attach to special chemically gated channels, or sometimes we call these ligand gated channels. And it's always going to be acetylcholine that is released to this skeletal muscle, and it will bind to these chemically gated channels and will allow sodium to move into the muscle cell. So similar to our neurons that we learned about, when sodium moves inside the muscle cell, it is going to create an action potential. Yes, we get action potentials through the muscle as well. So here's a nice little schematic that demonstrates this. Here's acetylcholine being released from our axon terminal, and it is going to go and open up those chemically gated channels that will allow sodium to move into the muscle cell. That action potential then, because we turned more positive, we reached the threshold, causes an action potential to propagate down the sarcolemma of the muscle, and it will move into the area of the T-tubule. That then will open up the calcium that is stored within the sarcoplasmic reticulum, and we can release that calcium to travel over to troponin, which will then shift tropomyosin. So let's go back to this to refresh our memories. Okay, so we will have calcium bind to troponin, which will shift tropomyosin out of the way, revealing the active sites, and our myosin head can then bind to the active site to cause the power stroke and cause actin to move in toward the M line, which results in a shortened sarcomere. All right. Now, after acetylcholine is released by the axon terminal, we have a special enzyme that can break it down so that we can get the muscle to relax. And that enzyme is called acetylcholine esterase. And it breaks acetylcholine down into acetyl and choline. We don't want acetylcholine to hang out around this cleft because then we would have unwanted extended muscle contraction. And the last thing I'll point out here is this is showing us an example of what's happening here at the motor end plate, where we have these chemically gated channels and acetylcholine is going to bind to it. That opens the channel and allows sodium to move in, creating a positive environment inside the muscle cell, which then causes an action potential to move down either side of the sarcolemma, the plasma membrane here. All right, and lastly, we need to talk about control of muscle tension. Our neural control initiates formation of cross bridges, and that pull exerted by the muscle is called tension. And the amount of force created by tension can vary based on the number of cross bridges that are going to form. Now, when we slow down our movement, we have more time to create more cross bridges and therefore can increase force. But something else that is important here is the ideal length of our sarcomere. So you could see that taking place here at the number 2, where we are able to create maximal tension when our thick and thin filaments are overlapping to the greatest degree. Now, when we have a sarcomere at rest and it is stretched past the ideal resting length, then we're not having proper overlap of the myosin heads and the actin filaments. And so we're going to have fewer cross bridges that are going to form. Whereas when we have a sarcomere that's shortened, like you see over here, then our thin filaments begin to overlap with each other, and that reduces the amount of space we have for those cross bridges to take place, and less tension will be produced. So when we have the proper length of our sarcomere here, we can increase the frequency of action potentials, or the number of signals per second that are being sent from the neuron to the muscle, to increase force a bit more, because that troponin will be flooded with calcium, allowing tropomyosin to move out of the way and the myosin head to attach to the active site. So that primary variable determining force production is going to be the number of myofibrils within the muscle that is able to receive that action potential from the neuron that controls that fiber.